Welcome to Norway, to the beautiful city of Oslo and to the Norwegian Academy of Science and Letters. We're here today with a very important announcement. One of the world's most important and prestigious science prizes. So to tell us the winners of the 2026 Kavli Prizes, President of the Academy, Annelin Eriksen.
Today, we are honouring groundbreaking work in 3 scientific fields. Work that has provided us with brilliant new knowledge, insight and ways of thinking about astrophysics, nanoscience and neuroscience. In the words of the founder of these prizes, Fred Kavli, the largest, astrophysics, the smallest, nanoscience, and the most complex, neuroscience. Without any delay, let us go straight to the winner of the 2026 Kavli Prizes. The Kavli Prize in Astrophysics for 2026 is awarded to Vasily Belokurov, Amina Helmi, and Rodrigo Ebata for uncovering the fossil evidence of past mergers, proving that the Milky Way galaxy was built through hierarchical accretion. The Kavli Prize for Nanoscience in 2026 is awarded to Eva Andre, Pablo Harillo Herrero and Alan MacDonald for foundational work that established the field of twistronics. And finally, the Kavli Prize in Neuroscience for 2026 is awarded to Christine Holt, Kelsey Martin, Erin Schumann and Oswald Stewart for the discovery of local protein translation in neurons and establishing its importance for brain development and plasticity.
Thanks so much, Annelin. And can I be the very first to congratulate all the winners? It's a magnificent achievement. Now, as you know, the Kavli Prize has 3 fields: astrophysics, nanoscience and neuroscience. The chair of each of the prize committees is going to present a citation, and then we'll get to find out a bit more about each laureate and their work in their own words. So let's jump right in with the biggest, astrophysics, and work that's changed our understanding of our galaxy. Chair of the astrophysics jury, Per Barth Lilje.
The Kavli Astrophysics Prize honors 3 astronomers, Vasily Belokurov, Amina Helmi and Rodrigo Ibata, whose work has profoundly changed how we understand our home galaxy and how galaxies like it came to be. For many years, a theory of galactic mergers and acquisitions prevailed. Massive galaxies grow through a long series of mergers, gradually swallowing smaller neighboring galaxies over cosmic time. It was a nice theory, but what was missing was clear observational proof. That proof is what the 2026 astrophysics laureates have delivered. Thanks to extraordinary data from major surveys, most notably the Sloan Digital Sky Survey and the Gaia Space Mission, the positions, motions and chemical fingerprints of billions of stars can now be mapped. By combining these vast datasets with innovative analysis and simulations, our laureates have revealed the Milky Way not as a quiet, orderly system, but as the dramatic survivor of repeated, violent cosmic collisions. Our galaxy can now be viewed as a richly structured archaeological site. Embedded within it are stellar streams and halo substructures, faint remnants of ancient galaxies torn apart and absorbed long ago. These stellar fossils allow us, even today, to reconstruct events that happened billions of years in the past. The smooth growth that had been envisaged was now shown to be a complex and turbulent assembly history. Vasily Belokurov demonstrated the crucial role played by ultra-faint dwarf galaxies in building the Milky Way. He mapped what became known as the field of streams, offering a comprehensive view of our galaxy's accretion history and its deep connection to the distribution of dark matter. Together with Amina Helmi, working independently, he identified the remains of the last major merger experienced by the Milky Way, the so-called Gaia-Enceladus sausage, characterized by stars on strongly radial orbits that make up a dominant component of our local halo. Amina Helmi showed that even the oldest merger events can still be detected among nearby stars through her pioneering use of chemodynamics, combining stellar chemistry with motion. She demonstrated how to distinguish stars born within the Milky Way from those brought in from outside. Her work revealed that the Gaia-Enceladus merger contributed a large fraction of the inner halo and likely heated the early galactic disk some 8 to 11 billion years ago. Rodrigo Ibata provided the first clear observational proof that the Milky Way is still growing today with his discovery of the Sagittarius dwarf's spheroidal galaxy. His subsequent mapping of his vast tidal stream and his groundbreaking work on the rich and asymmetric halo of the Andromeda galaxy gave astronomers vivid visual proof of how galaxies are surrounded by sprawling debris fields, evidence that galactic formation is an ongoing, dynamic process. Together, these three scientists have transformed the Milky Way into a readable record of cosmic history. They have shown us that galaxies grow through hierarchical accretion, that the scars of ancient mergers remain visible for billions of years, and that our own galaxy bears the unmistakable imprint of its tumultuous past.
Thank you, Per. It's awe-inspiring work. And here are those three laureates, Vasily Belyakurov, Amina Helmi, and Rodrigo Ibata.
We have discovered that the Milky Way, the galaxy we live in, experienced many mergers with smaller objects a long time ago throughout its lifetime. The biggest one that happened 10 billion years ago, long before the Sun had been born.
One of the structures that we found was the Sagittarius dwarf galaxy. So this is a galaxy that's currently being swallowed by the Milky Way, leaving a huge trail of stars behind it that completely envelops our galaxy. And we've also been finding similar structures around the nearby galaxies that inhabit our local group. The galaxy that we worked on in particular is called the Andromeda galaxy. And there again, we saw this, the accumulation of smaller galaxies being swallowed up by the larger galaxy. What this tells us is that the galaxies form in a hierarchical way.
This is important for our future attempts to understand the emergence of structure, emergence of first stars, of life, of first galaxies in the early universe. We will be able to reconstruct the evolution of the Milky Way and rewind back time much better now. We will also be able to understand the dark matter by mapping the dark matter distribution in our own Milky Way in unprecedented detail. And we now know that the dark matter halos of galaxies like our own are much more complex than we previously thought. So we have prepared ourselves for this quest in the future.
Like I said, awe-inspiring. Now, from the vastness of the cosmos to the smallest of the small, nanoscience. Chair of the Nanoscience jury, Mariam Einarsrud.
This year's award recognizes three scientists, Eva Andrei, Alan McDonnell, and Pablo Jarillo Herrero, whose pioneering foundational discoveries revealed how a simple geometric twist between atomically thin layers can dramatically change a material's properties. Twistronics, as the field came to be known, quickly became one of the most powerful platforms in condensed matter physics. It was in 2009, using scanning tunneling microscopy and spectroscopy on suspended twisted bilayer graphene, that Eva Andrei and her group first showed that small variations in twist angle profoundly modified the electronic structure. She identified a special twist angle, later known as the magic angle, where electrons form unusually flat bands. These flat bands enhance electron interactions and lead to new collective states of matter. The demonstration of flat bands can be engineered through geometric control rather than chemical composition, represented a fundamental advance in materials design and marked the birth of a new field now known as twistronics. In 2011, André's groundbreaking experimental work was placed on a firm theoretical foundation by Alan McDonnell. He developed a continuum model describing how interlayer twist reshapes the electronic band structure of twisted bilayer graphene. This framework quantitatively explained the emergence of flat bands and has since become the theoretical foundation of so-called Mori materials. Subsequent technical advances by others enabled precise control of twist angle, carrier density, and dielectric environment, completing the experimental toolbox of twistronics. The broader significance of twist-engineered flat bands was demonstrated in 2018 when Pablo Carrillo Herrero's group observed correlated insulating phases and unconventional superconductivity in magic angle-twisted bilayer graphene devices. These results show that superconductivity can emerge in a system of two weakly coupled graphene layers when tuned by twist angle and electrostatic gating, opening a powerful new platform for exploring interaction-driven quantum phases. The use of geometric alignment as a turning knob for engineering the band structure introduced a new paradigm in nanoscience. This has led to a wealth of new phases of matter and new quantum behavior such as unconventional superconductivity and correlated insulating states. Twistronics has opened a rich landscape of quantum phenomena and continues to shape research across quantum materials and nanoscience.
Thank you, Marianne. We've become familiar with the way that materials change their properties at nanoscale. Now we know that adding a twist and a magic angle takes us into a whole new world of possibilities. To tell us more, let's hear from laureates Eva Andre, Alan McDonald, and Pablo Carrillo Herrero.
Here at the University of Texas, we realized that twisting 2D materials could create a new platform for making a flexible class of artificial materials in which nearly every property known to man could occur.
When you take two graphene layers, you put them on top of each other with a twist. The electrons that are inside, their kinetic energy is going to be quenched, and then they can start seeing each other and collaborating to create new states of matters like magnetism or superconductivity. This shows that you can create new states of matter without chemistry, without changing the chemical composition of a material, just with a twist.
Because with twistronics, we can change the properties of matter at will, and we can actually make one material behave like multiple other materials. This is something that is extremely flexible in terms of technological applications, because now, with very simple ingredients, you might be able to realize a variety of behaviors that you need for different technological applications, which range from special quantum technologies like quantum computing or quantum sensors to new types of devices that can compute the same way the brain computes. These are neuromorphic devices which are being investigated for artificial intelligence. So in principle, we have now a very broad spectrum of applications for which we might be able to use a simple set of materials because we can change their properties by playing with geometry.
Who'd have thought that a simple twist could do all that? And now finally to neuroscience and research that, while showing how nerve cells work, revealed much more about how the brain works. Edvard Moser, Chair of the Neuroscience Committee.
Neurons are unusual cells. Their central body is small, while most of their volume lies in long branching axons and dendrites that form thousands of synapses or contacts with other neurons. These synapses operate with striking independence, each relying on its own supply of specific proteins to function and adapt. Genetic instructions for making those proteins, however, are stored far away in the nucleus of the cell body. This raises a fundamental question. How can a neuron deliver the right instructions to the right synapse, sometimes at great distances, without creating an overwhelming internal traffic jam? For decades, no one knew how this logistical puzzle was solved. Through pioneering and complementary work over several decades, the 2026 Kavli Prize for Neuroscience laureates Christine Holt, Kelsey Martin, Erin Schumann, and Oswald Stewart showed that neurons could make proteins exactly where they are needed, whether in axons, at synapses, or in dendrites, without needing to rely on the cell body to the extent that many other cells do. This insight came to transform our understanding of how the brain develops, adapts, and stores information. Oswald Stewart provided some of the first evidence that this local control was enabled by local protein production. In electron microscope studies of hippocampal neurons, he demonstrated clusters of ribosomes at the base of dendritic spines and in postsynaptic sites. This observation led him to suggest that synapses might be able to make proteins on-site, linking local protein production directly to synaptic activity. Local protein synthesis is essential for the growth and guidance of developing axons. Christine Holt demonstrated that the growing tips of axons, known as growth cones, contain a rich collection of messenger RNAs, suggesting that proteins could be assembled locally. Local synthesis would enable nerve fibers to respond rapidly and precisely to guidance signals during brain development. Kelsey Martin's work showed that separate branches of the same neuron can regulate synaptic strength independently through local translational control. Remarkably, she was able to visualize this translation in live neurons. Her work helps explain how learning and memory are stored in the brain with such exquisite specificity. Erin Schumann showed that messenger RNAs are translated locally in dendrites, both under normal conditions and in response to synaptic activity. Without local protein assembly, the long-lasting synaptic plasticity at synapses in the hippocampus would not be possible. She also developed powerful tools which identified and visualized newly made proteins, opening the way for modern large-scale studies of how proteins are synthesized and degraded across the cell. Together, these discoveries have shown us that neurons can control protein production with extraordinary precision in space and time. They tell us how axons find their way, how individual synapses change with experience, and how long-lasting brain plasticity is sustained. This research has been fundamental both to neuroscience and cell biology as a whole.
Thank you, Edvard. Now, this is research that has great potential for human health and that spanned several decades. Telling this story in their own words, laureates Christine Holt, Kelsey Martin, Erin Schumann, and Oswald Stewart.
I've been working all of my scientific life on the question of how the brain gets wired up properly in development. How is it that neurons make connections with the right neurons?
Prior to our discovery, it was thought that all the proteins were made in the central part of the neuron, the cell body, and shipped out, a central factory, sending out trucks.
But what Ozzy, Christine, Kelsey, and I showed is that the machines and the messenger RNAs, the templates for proteins, are actually localized explicitly out in the dendrites and axons, where when synapses are activated, proteins can be made on demand and can give rise to changes at synapses that store information, like memory.
So each neuron has a single cell body and a single nucleus, but can form thousands of synapses. By being able to change the structure and function of individual synapses, that massively enhances information processing in the brain. And understanding how that happens through the local translation of RNAs at the synapse, enables us to have a deeper understanding of how the brain functions.
And our hope in the future is that these basic findings will help with therapies.
And perhaps how synapses can actually be regrown after injury.
And there you have it, the 2026 Kavli Prizes. Extraordinary science that's changed how we understand our home galaxy, told us how our brains work, and helped create a new era of materials for a quantum future. Science is amazing. Congratulations again to the laureates, and a particular thanks to the committee members who come together from all over the globe to help find the best of the best. The laureates have got a busy time ahead of them, and we'll be meeting them again at the prize ceremony on Tuesday the 1st of September at the Oslo Concert Hall. Until then, goodbye.